All right, hi everybody. Thank you for sharing your time with me today. My name is Connery Meyer, and I'm going to be presenting some work that some colleagues and I in Shimeng Tan's lab at UC Davis have recently published. The title of the work is Holistic Engineering of Cell-Free Systems Through Proteome-Reprogramming Synthetic Circuits. And I know it's quite a mouthful, but I'm going to try and unpack it such that it makes some sense. So to begin, I kind of want to give somewhat of a background on synthetic biology such that it contextualizes why we did what we did. So kind of the current mantra of synthetic biology is this idea that we can insert synthetic DNA, which codes for RNA or proteins that are useful to us, and then we can trick the cells into reallocating their materials and resources to produce the protein of interest. So an example of this could be we can put in DNA that encodes for insulin such that we get these cells in large batches to produce insulin that we can purify and give to diabetic patients. Now, these systems can get arbitrarily more complex where we can modify it such that it doesn't just produce the insulin or whatever the protein of interest is all the time, but that it does it inducibly, so off of certain signals. These signals could be changes in pH, increases in certain molecular or molecule concentrations, or external signals like ultrasound. And then these systems can be linked together such that only when both signals are present, they can lead to the final production of proteins. Those can be used in bacterial therapeutics, where it can be sensing for pH, such that when the cell arrives at the tumor and experiences the drop in pH, and say a signal of ultrasound localized to the tumor site leads to the production of some therapy or therapeutic that then destroys the cancer. Now, because these systems can get so complicated and the underlying biology is so involved, we typically abstract away all the complexities and kind of draw it all as these circuit diagrams. So we just sort of assume that these circles or nodes are the reactions and the fluxes are sort of the transitions from one to the next. So that could be the production of a protein that then moves to the next step. And so we draw them as these perfectly insulated systems that just kind of get inserted into these cells. I think it's pretty helpful to remember that these cells are actually incredibly complicated. They house thousands of different molecules, have hundreds of different reactions happening simultaneously. They're somehow all housed within these, single femtoliter volumes, and that are able to replicate once every 20 minutes. And so, as you could probably imagine, there's a lot of cross interactions. So the cell does not recognize these arbitrary lines that we draw around our synthetic circuits. And so what we see is that there's the same resource limitations that our system, that our synthetic system will incur, as well as the rest of the cell. And then it's also subject to the inherent stochasticity within these cells as they go through their different cell cycles and changes in available resources. And we also see that there's a lot of reactant and product catabolism where the cell is just pulling whatever it needs. And so if you want to use amino acids or certain molecules, they'll oftentimes be redirected into other pathways. So what we advocate for instead is, instead of trying to ignore all these other complexities, we want to integrate into them. And so this could take the form of increasing the reactant concentrations, minimizing degradation pathways, and eliminating other processes that might pull away from the available energy. So I've been pretty abstract about what these systems actually are. And so I kind of want to dive a little bit deeper and pull away some of that abstraction. And so really what we're talking about is protein translation. And so here's a really simple diagram of what that is, which is essentially the process of taking amino acids and producing the protein of interest. So we can make some RNA, which then guides as a template for the ribosome to know how to stitch these amino acids together in certain sequences to then produce the protein that we care about. Now, as many of you probably are familiar with this process, it occurs naturally in pretty much all cells, including ours, all the way down to bacteria. But something that's interesting is that we can actually purify out a lot of these components and then reconstitute them outside of cells. Now, the reason why you might want to decouple the act of translating proteins from cell viability is that it allows you to express toxic proteins. So things that would otherwise kill off cells can now be produced. They can also allow for the encapsulation inside of artificial membranes to create cell mimetics. They can be dried down into paper and then transported, eliminating the cold chain for paper-based diagnostics. And because they can be arbitrarily scaled and they're not encapsulated, they're highly amenable to high throughput screening in combinatorial studies. And so I just kind of want to look at the actual process here, so it's not this weird black box. And so essentially what we do is we take our starting strain, and we inoculate a big culture, grow up tons of these cells, and then pop them open. And then we harvest their cytosol such that all the proteins that were present inside, including all of our translation factors, are still present. And then with a couple extra preparation steps and reconstituting a specialized buffer, we can then reconstitute protein translation, as is shown here, along with all the translation factors needed to make that happen. And so the point of this work is to increase this flux. So we want to be able to take more amino acids and produce more of our target protein. Now, given the utility of the system, as you might guess, we're not the first people to try and improve this. So generally the strategies that people have taken is the removal of harmful proteins. So this can take the form of proteases, nucleases, or other catabolic enzymes, such that in the final whole cell extract, there's less things pulling away from the actual products that we want to use. The other option has been to make the cell-free reaction and then just supplement in things that would be useful. So this can include chaperones, increase the folding of the final protein, or transcription and translation machinery, which then aid in increasing the flux through the pathway. Now, while these systems have provided some benefit, typically they're fairly marginal and they don't scale very well. So it's very difficult to do genome engineering at a larger scale, and the addition of transcription and translation machinery require the preparation of tens of proteins, which makes it very difficult to scale. And so what we wanted to do instead is look at actually engineering of the cells such that that is a much more scalable approach. And so what we did was we took all the translation proteins, translation factor proteins, and overexpressed them. And so this includes 11 different proteins that we spread across 7 different strains. And so by doing this, when we lyse the cells open, the local concentration of all of those translation factor proteins is now greatly increased. And so now we have all more proteins available to then force translation to happen. But what we found was that while we did this, we also created this massive proteome reprogramming. And by causing all these changes in the rest of the proteome, we see that there's actually these other beneficial effects that then further increase the flux through this pathway. And so to kind of illustrate that this system actually worked, I wanted to kind of show comparisons of it to other systems or other whole cell extracts. And so in here, what I'm doing is I'm comparing these extracts based on their ability to produce DE-GFP, which is essentially a truncated version of eGFP shown to work well in cell-free systems. And so I'm comparing it to BLP, which is the strain that we're using just carrying plasmids without any coding proteins, or the wild type strains, so not holding anything else, and then a commercially available S30 extract. And so what we see is generally this 2 to 3-fold increase in total efficiency. And this is then further illustrated in our semi-continuous reaction, where we have like a membrane that allows for diffusion of harmful products from the reaction and then more reactants into our cell-free reaction. And then we also scale these reactions down and then ran time courses, and this difference is even more enunciated. And so what we wanted to do is we wanted to try to understand if the changes that we made intentionally are the sole cause of this enhancement. And so to test that, what we wanted to do is we took a normal extract. So this is just our strain without expressing any proteins, and we lysed it open, and then we purified out all 11 proteins that were necessary or that were present in our modified system. And so by supplementing them together, we wanted to see if we can recreate what our whole cell extract was. And so here on the x-axis, we're increasing the translation factor protein mixture, and on the y-axis, we're seeing the resultant protein that we're making. And so what we see is this concentration-dependent increase in synthesis capacity based on translation factors. But even by the time that we get closer to 20 micrograms, which is well above the amount that was present in our whole cell extract, we're still only barely getting to half of the benefit that we were getting in our system. And so what that told us was that there was something else happening that we weren't accounting for. So to get a better or more holistic view of how our system was working, we wanted to switch to proteomics. And so because our system was pretty complicated and we have a lot of different facets, so to try and understand what our system was actually doing, we wanted to break some of these different variables down to see sort of how it affected the proteome. So to start, what we have is BLE or BL21, which is just a wild type strain. So we're not doing anything to modify it. Our next one was to see if just by expressing some protein, doesn't matter what that protein is or how it affects the rest of the system, we would change the proteome. And so we decided to express CFP or cyan fluorescent protein because it should affect nothing else in the system. And our next one is a single strain from our full 7-strain set, which is the one that is most abundant. And so it's producing EF2 and EFTS. And then of course our final strain, which is all 7. Now to kind of illustrate the differences in expression capacity, we once again see this 3-fold increase in our strain, our 7 strain versus all 3 other ones. Now, being as proteomics data can be pretty difficult to interpret, it can oftentimes be misleading. I want to start off with looking at the individual replicates to give some credibility to the rest of the stuff that I'm going to show you. And so to do that, I'm looking at the principal component analysis, or PCA analysis, of all the individual replicates, which is looking at all 2,000 different proteins that we identified and all their concentrations in all the extracts. And so what we see here is that all of the extracts group tightly together, as we would hope, given their replicates. And these ellipses that are surrounding them are our 95% confidence with a t-test. And so we expected that they would all be fairly separated, and that the single strain and 7 strain would overlap, which we do see here. And so now we're going to move to averaging all these replicates and looking at the changes in all 2,000 different proteins. And so this volcano plot can be kind of confusing, and so I want to kind of break that down a little bit more, where the x-axis is the fold changes between the noted extract and our baseline. So for BLCFP, this would be how much protein X increased in comparison to our wild type strain. And so as you go farther to the right, it's a further increase in the protein concentration. And on the y-axis, what we're showing is the significance of that change. So this is once again a t-test comparing the 2 strains. And so as you go up, you get increased confidence. And so what we did was we set thresholds of a 15 or a 25% increase and a 95% confidence to then showcase the those that had increased and those that had decreased. And so what we see is that CFP is pretty evenly split between increase and decrease, but our single strain and our 7 strain is massively decreased. And so to kind of understand where the distribution of proteins went, we look at the percentage of the total protein. And so we group the proteins based on their gene ontology. And so when we look at this, as we go farther to the right, so closer to our main system, we see that there's a massive increase in our gene expression, and that gets compensated for by metabolism and homeostasis. And so when we kind of break these down into subcategories, we see that most of the components in gene expression that are contributing to this increase is our translation factor proteins, as we would anticipate. And the decrease is primarily in metabolism, mostly amino acid metabolism. And so if I were to plot this sort of on our original diagram, we see that we get a decrease in amino acid catabolism, which is going to increase our availability of amino acids. We're decreasing secondary metabolism, which allows us to have more available energy as we're using it less on extraneous processes. And there's a decrease in degradation, which is allowing for a higher accumulation of our target protein. We also see an increase in ribosome biogenesis, which is ideal. And so we want to just kind of demonstrate that we can use the system on other proteins, such as ferritin, which forms these nanocages. This can be seen in the SEM. And we see that we can make Cas9, which is a much larger protein, which is generally very difficult in E. coli cell extracts, showing that we can get almost a 5-fold increase over commercial strains. Now, let's kind of wrap up what we talked about here. So we show that we can demonstrate plasmid-based approaches to functionalizing cell-free systems, which up to date has been actually detrimental. And so we're kind of showing that this is a new way in which we can further improve cell-free systems. We also show that we can reprogram the proteome without any genetic manipulation to the genome. And we showcase that you can work to integrate our orthogonal circuits along with the cell physiology in order to create this sort of like holistic synergistic effect between our system and the cell as a whole. Now, as we move forward, what we want to do is we want to really understand how all these changes are being made. And so we want to look at the feedback loops in our holistic approach and identify what in those changes are the most beneficial. Now with this holistic approach, because now we can produce a lot more protein, this is useful for incorporation of unnatural amino acids and new proteins, making various ribosomes and expressing complex, bigger, and more complicated PTM proteins. And so with that, I'd really like to acknowledge my lab, specifically Dr. Chiming Tan for his guidance, and especially my co-author, Luis Contreras-Lano. And so with that, I'd like to invite everyone to join me for the Q&A session, which is going to take place on Thursday at 8 a.m. So thank you very much for your time, and I look forward to any and all feedback that you would like to share with me. All right, thanks so much.